All right, so hopefully that kind of makes sense to you guys. We're jumping to a whole different thing here to a certain extent. It's actually not completely different. But selective permeability, your membranes in your cells, in all cells, are selectively permeable. They allow some things to cross, but not all things, right? So you need certain things in your cells. So obviously things need to be able to move through the cell membrane. But different things move across that cell membrane differently, right? So if I redraw a picture here, I'm not going to draw the whole complicated thing all over again. And this is not a different page. You could draw it in your notes, just put it in a separate place. If I have a membrane, again, I have these, let me just go ahead and use blue. I have this lipid bilayer, right? So that blue that I just drew, those two blue lines, those represent those polar head groups of those lipids, those phospholipids we were talking about, right? Inside, in between, let's make that a little bit smaller. Inside, in between, this whole portion of the membrane, again, is very nonpolar, right? So what we're talking about in terms of transport, right? Small nonpolar molecules can cross the membrane by themselves, right? In order to get through from one side to the other, a molecule would have to be very small and nonpolar. And luckily, molecules like oxygen that you need in your cells are small and nonpolar, right? Oxygen can be dissolved in water, so it's actually okay. It's in the water and you can transport it to your cells. But if it wasn't small and nonpolar, there's no way you could get it through the cell membrane to get into your cell. So we're assuming this is the inside of the cell, right? This is the outside of the cell. But you need oxygen in, and you need to have that happen without any trouble, right? Oxygen can go directly through the membrane because it's a small nonpolar molecule. And not only do you need oxygen, but you actually have to get rid of waste products in your cells. You're going to make carbon dioxide as a result of respiration, also a small nonpolar molecule. It can travel directly through the cell membrane. So it can get out of your cells without any issue, right? That's actually really important. These two molecules have to move in and out of your cells, but they're small and nonpolar, so they can go directly across the membrane easily. Polar molecules, on the other hand, don't go through easily, right? If it's a polar molecule, like to a certain extent, water, right? Water is not going to pass through this membrane very easily. So like I said before, right, it doesn't pass through the membrane very often, very frequently, directly. It actually usually goes through a pore that's opened up in your cell membrane. So you would have a transport protein that allows water to go in and out of the cell, right? So small polar things need to go through proteins, channel proteins, to get from one side to the other. Again, it could be something like water, right? That's polar enough that, again, the hydrophobic tails don't like water at all. It's going to need to go through a channel as opposed to going through the membrane directly. But you could also have things like electrolytes that are in your cells, like potassium ions and sodium ions and chloride ions, right? That need to be able to make their way through. Now, they're not polar molecules technically, but they're ions that have charges, and they're going to behave much more like a polar molecule like water than like the non-polar parts of these tails, right? They can't go through directly either. So if they're going to travel through the membrane, they need to go through a channel protein, right? Whatever direction they want to move, right, depends on the concentration of those things inside, outside of your cells. But they can move through those transport proteins. Otherwise, larger molecules have to be moved in vesicles. Right? And we're going to talk about that later, right? Endo and exocytosis, so I'll show you what that would look like later. You are supposed to come up with another example of a semi-permeable object, right? You can come up with whatever you want because you actually should know some of these things. Something that allows certain things to go through but doesn't allow others. Right? So just an everyday object, that's a good example. what can pass through it, what can't. We can check on this later to make sure you have something good, but go ahead and try to answer that yourself. But otherwise, you should know how things can go through the membrane, right? If it's small and nonpolar, again, these two molecules are nonpolar, so they actually don't have any trouble going through the nonpolar part of the membrane, whereas this polar molecule can't go through the red. These things are essentially polar, right? In terms of they have charges, actual charges. Those ions are just like polar molecules. They have to go through those transport proteins as well. Otherwise, this last portion, right, receptors, there are going to be a couple different kinds of receptors in your cells. So these are proteins that detect a signal and act because of it. So you're supposed to be able to control your cells. You're supposed to be able to tell your cells what to do, right? You have this body that has lots of different tissues and cells and organs and everything else. And every cell has a specific job, but those cells do not necessarily do things without being told to do them first. So you have these ligands, as we're going to talk about, right? But essentially, you have a lot of different signal molecules in your body. So we're starting out talking about a ligand that is a signal molecule, essentially. But you have other kinds of signal molecules in your body. You have hormones in your body that affect cells and make them do different things, right? But a ligand is a specific kind of signal molecule that combines with a receptor to make an activated protein that makes your cell do some function, right? So this is just a specific kind of way that your cells can be communicated with. One cell can tell another cell what to do, right? But there's different kinds of receptors. There's intracellular receptors and there's membrane receptors. So I'm going to draw two pictures here, right? So this is just a division line, right? So to separate one side from the other. But an intracellular receptor, again, I need to draw kind of a membrane. And again, I'm not going to draw all those phospholipids every time. It doesn't make sense. But I always make two lines because then I have my bilayer, right? You can still kind of see that, understand that. But this is one side of the membrane. This is the other. I'm going to have a ligand molecule that, again, we just need to have some kind of protein here, right, that has a certain shape, but this is my signal molecule. This is my ligand. It's supposed to be. So label it down there appropriately. I'm just going to highlight it so you can tell what's what. But this ligand is attempting to find a receptor. If it's an intracellular receptor, that means it's on the inside of the cell. It's inside of the cell membrane. Intra means in, right? So in order to get this receptor, in order to get my ligand to my receptor, to activate that receptor, this molecule needs to go through the membrane, right? Once it is across the membrane, you can combine this thing and this thing to make a new structure. Again, we do pictures like this all the time that may or may not make sense, but I essentially have this receptor protein that changes when it's associated with a ligand. So I'm going to make a square shape with that triangle notch because it's now got the ligand inserted inside of it, right? So I got that signal molecule all the way to my receptor protein, right? The receptor protein itself, I'll just make it blue, right? Changes shape, and that new shape gives you some new function. It'll now do something that it didn't do before, that whatever purpose it has doesn't really matter, right? Something will now happen in the cell that wasn't happening previously. So again, this protein is essentially off until a ligand molecule attaches to it, and then it'll turn on and become active and do whatever it does in the cell. But this is an intracellular receptor because it's on the inside of the cell, right? A membrane receptor is different, right? A membrane receptor would actually be built into the membrane. So I got my protein again, my receptor protein that is embedded in the membrane itself. So I still have outside, inside context. But the difference being, in this case, if I got a ligand molecule that needs to get into this receptor, right? Excuse me, I used pink for that. Let me try one more time, right? So if I want that ligand to enter that receptor molecule, right, it's a little bit easier this time because there's a difference between this picture and this picture, right? In order to enter the receptor here, the ligand does not have to cross the membrane, right? So if I go from one shape to the other to activate this thing, again, this is still my receptor, it's just my activated version. Maybe I should use like a different color for that, right? The activated one is the one that has changed shape and now has a different function, a new shape, a different function. And that happens because of my receptor molecule, right? My receptor molecule, excuse me, not receptor, I keep saying the wrong word. Ligand molecule has entered my receptor. Different colors all the time, my bad. Thought I clicked on the pink, but I clicked on the green. Right? My receptor has a ligand associated with it right now. That ligand has attached itself to the receptor molecule and now activates that protein so that it does whatever it's supposed to do. Now, obviously, it can do its job on the surface of the cell. It doesn't have to be inside the cell to do its job. But there's a difference between intracellular and membrane receptors. And the biggest difference is whether the ligand has to cross the membrane or not. Because what I would ask you about this on the test probably is which one of these things do you think would be harder to activate? What kind of receptor would be harder to activate? An intracellular receptor or a membrane receptor? So think about that. You might want to jot down an answer. I might ask that question again in class, but I'm not going to tell you the answer right now. You should be able to kind of understand which one would be harder to turn on. Because I told you the major difference, right? So just think about that, figure that out. Otherwise, you got a little bit of work to do associated with this section on Google Classroom, so get that taken care of as well.